All right, here we go. This is a tough lecture. It is, I forgot to fix that again. It is the fall of 2020. It is week 2, and this is the oogenesis. This is just a tough lecture, I think. And I will assume everybody's had mitosis and meiosis before, so hopefully you know a little bit about that because I can't teach all of that because this is hard enough. Um, all right, so how do we make an egg, basically? We did a sperm last week. That wasn't too bad, I don't think. But the egg is not an easy subject. Very confusing, like this little guy here is confused about something. And he may be studying embryology already, and that's probably the look some of you are going to have on your face after we get done with this lecture. But anyway, it's always a bad sign when the authors are confused and the authors are not on the same page. So, yeah, and you're probably going to be rewatching this, I would guess. So you make sure you go to the Amoeba Sisters on YouTube if you're shaky with meiosis. Now, meiosis is a little the way they teach it. There's a little twist, I think, to it. I'm not sure because we're going to actually get stuck. We're not going to make it all the way through meiosis in this story. We get stuck twice as we go through this, which is very strange. Here's the players. The story takes place within the ovary, and we don't really have much of an ovary, right? We're still kind of talking about the formation of the embryo. But this is where the story will take place. This is the mature, of course, ovary. Oogenesis, pronounced oogenesis, oogen like 10, like egg, oogenesis, not oogenesis or oogenesis, oogenesis. It takes place in the ovary, the story, and it starts, we actually have to jump ahead of where we are because we haven't even got to, we don't even know what the trilaminar disc is yet. But in week 4, we get a folding of the embryo. So this is kind of going into the future. And we haven't even created a yolk sac yet. But where does the story start? It starts basically with the yolk sac. And the tissue that's is destined to become an egg is actually has a name. It's called the primordial germ cells. And about week 4 or so, these primordial germ cells, they are born in the wall of the yolk sac. Actually, it's the secondary yolk sac. And they stream out of that wall and migrate to a location on the gut tube, which we'll talk about when the time comes. And there in the gut tube, they start growing in numbers by mitosis, not meiosis yet. So they start splitting and building up a huge population right here. And so here they are, and they kind of hang out right there. They also induce. What does induce mean again? Induce means that they secrete. They go through a central dogma, they make a protein, probably a growth factor, and they release it. And that growth factor soaks into the cells around them and causes a change. It turns on the central dogma in those cells. That's induced. But it causes the surrounding tissue to turn into support cells, and that's all I'll say. We won't go down that rabbit hole. But those support cells are there to kind of nurture them and help them in their growing process. And they're formed, eventually the growth of these cells gets so big that you have a visible bump that you can see under a microscope. And that has a name, of course, and that's called the genital ridge. And at this point, it could be male or female. It could go either way because germ cells could turn into spermatogonia, or they can turn into our primordial kind of egg, if you will. But in our example, of course, we're going to go down the female track. So these germ cells in the ridge will actually, some of them, they're still growing, but some of them will actually create the ovary itself. So the ovary is created from these primordial germ cells, and they keep growing themselves. So they've kind of created a case for them to grow in, and that's going to be called the ovary. Okay, meet the oogonia. So the primordial germ cells will continue to divide and mature and divide and mature, and at some point they are large enough and there's enough of them that we get a name change. So we call them now the primitive eggs, aka oogonia. Oogonia. So that's the end of the primary germ cells once they reach a certain size and maturity. Probably around the second to third month this is happening of development. And by the end of that third month, they're still dividing, but now the key is the oogonia no longer divide by the process of mitosis. They divide by the process of meiosis. So they start dividing via meiosis, and all of them do. And there's millions of them. And they divide through meiosis. They get through, you remember the first phase of meiosis is interphase. And so they replicate their DNA, right? So now there's two strands of DNA. And then when they go into prophase, that's, remember the saying IP mat is how I remember it. Prophase, metaphase, anaphase, telophase, cytokinesis. When they just start the process, they thicken and they become visible. In prophase, they stop. They become arrested. So that's kind of weird. So all of these millions of oogonia, they start prophase and then they shut off. And it happens right after the genetics from mom and dad mix. So crossover, which also happens in prophase 1, has completed, and then they just stop and are frozen. And that has a name. That's called meiotic arrest. And if you really want to get into the weeds on this, I think that's good enough for embryology class. But if you're in molecular biology or developmental biology, you probably know there's several phases within phases. So they really get stuck in what's called diplotene stage of prophase 1. That's where they get stuck. But I think that's going to be too deep. If you just know they get stuck in prophase 1, that's going to be good enough. All right, but crossover has occurred. But nevertheless, so now we get a new name because they've crossed over and their genetic code is mixed up amongst the cells. So now they become a primary oocyte. A primary oocyte. But the primary oocyte is not going to become a secondary oocyte for quite some time. In fact, we'll almost be done with this class and we'll get into a secondary oocyte. Here's an overview. So this is a good study guide. It wasn't drawn out perfectly, but here's kind of where we were so far. So we had some primary germ cells, which are 2n, by the way. Everything's 2n. We haven't went far enough to reduce the number of chromosomes. But those formed in oogonia. That entered meiosis 1, went through interphase, just 1. Remember, interphase, the chromosomes are so small and they're unwound and they're unruly. And by the time prophase 1 comes, they're thick. They have thickened into, quote, chromosomes, the ones that we can see. But then right after crossover, it stops. And that's where we're going to be stuck for a long time. Jumping ahead, we are going to wake up and we're going to go through metaphase, anaphase, telophase, and we're going to create a secondary oocyte here and a polar body. And then we're going to start meiosis 2, but we're going to get stuck again at the end of the story in metaphase 2. And if you don't get pregnant, your eggs will never, ever progress past metaphase 2. When you have your period, the egg that flushes out is stuck in metaphase 2. The only time your egg will actually finish meiosis is if it gets a sperm inside it. If you get a sperm inside there, it wakes up the meiosis 2 again, and it goes through anaphase, telophase, and then you have a mature And we will talk about the next week. We'll talk about that. But then you'll have one mature egg, but that egg will have a sperm in it. So that's kind of the big picture where we're going. And there's another way to look at it. Remember, there's 3 polar bodies that are made, which don't do anything. If the sperm actually gets inside of here, you'll abort. The fetus won't be viable. You only make one good egg out of this whole story. So why does meiosis stop? And I'm not sure if they know the entire story on this. Carlson goes into it a bit. So it's believed that this stopping in prophase 1, or this meiotic arrest, occurs when the oogonia gets exposed to 2 types of chemicals or molecules. Our friends cyclic AMP and cyclic GMP, or cyclic adenosine monophosphate, cyclic guanosine monophosphate. So kind of nucleotides here are involved. And the specifics of the reaction are not well understood, and they're theoretical, so we won't even have to go into those. But no, it's when these 2 molecules get pumped and start to build up inside the nucleus, it shuts off it puts things into meiotic arrest. And some of those primary oocytes, they can be dormant. You females, they're dormant inside you right now. They're just waiting their chance to awaken. And then we'll talk about the great race for ovulation, because many of them, the chances of one of these actually ovulating, getting out of the ovary, is pretty slim. But some of these can stay suspended for 5 decades, because females in their 40s can, before, well, their eggs are still viable even into the 40s. So they can wake up, but they've been asleep for sometimes 5 decades or 40 years. So that's crazy. So the question then, I know you're thinking, some of you who are awake are thinking, well, how can they do that? How can they sleep so long? And don't they get damaged? And that is a problem, right? You probably know that. Having a baby in your 40s is a little risky for genetic defect like Down syndrome in particular. And that's because these primary oocytes have been asleep for years and years and years, and they've been exposed to radiation. You may be flown across the country. You've had an x-ray at some time. So all those years increase the chance of chromosomal damage. But they still can wake up. What protects them when they're sleeping? They do have some protection while they're sleeping. So they accumulate a fluid inside of the cytosol, and we'll just leave it at that. We won't get crazy into that. But there's a fluid that accumulates in the nucleus. The nucleus swells up pretty good, and that fluid gives them some extra protection as they hibernate. And then we already said that if you try to have a baby in your 40s, you have a much higher chance of having some type of birth defect or a Down's baby, because of your lifetime exposure to teratogens, which we talked about last time, like cigarette smoke, pollution, just radiation from life, right? You broke a bone, you had to have an x-ray. You've flown, you get radiation flying across the country. Meet the primary follicle. So about the 4th month of development, a new layer starts to form around our primary oocyte. So right now we have this naked primary oocyte hanging out. So we need to make a follicle out of this, and that's the next step. And basically it's a basal lamina, so you have a follicle right here, the primary follicle, and you're going to grow a single layer of cells around it. So we're working our way to making a follicle, because this thing has to hatch. This has to hatch out of the ovary. Every month you have one of these hatch, but it's not ready yet. We got to build it first. So we get a basal lamina built. And basically this is, for right now, it's made of flat cells, simple squamous cells, which have a name, of course, and they're called follicular cells. after the thing we're trying to build now is a follicle to further protect this primary oocyte. So we have a single layer of follicle cells. Some authors call these granulosa cells because as they multiply and as this layer gets 2 layers and then it gets 3 layers, it gets thicker and thicker. Eventually it's going to become the granulosa layer. So it'd be nice to call it granulosa cells, but unfortunately the chiropractic board books call them follicular cells. And Carlson calls them granulosa cells. Kind of the European books tend to call them that. And then we could also say, oh, well, just like endothelial cells make up the endothelium, we could say these follicular cells make up the follicular epithelium, which is only 1 layer thick at this time. And at this point, our job is to grow like about 20 layers here and make this really, really thick. And so you could say that the process of folliculogenesis has now begun. Folliculogenesis. And now we have a name change because even though it's only 1 layer thick, we've created a new structure, so we have to name it. So the combination of this single follicular layer and our primary oocyte together, this is called the primordial follicle. Primordial follicle. It's not very tough yet. It's only got a single layer, but we're going to change that. And these guys are really, really delicate. So by the 5th month of development, still in your mom's tummy, there's 7 million primordial follicles in this growing, inside this primitive ovary, which itself is still growing. But for reasons they don't understand, these things die like crazy. So by the time birth comes around, that number is down to 2 million. So you've lost 5 million of these primordial follicles have degenerated and died off already. But that's still a lot. You still got a lot of these things, and they're much thicker by this time. And then, following on this thought, by the time puberty, because these primordial follicles, they're going to change into a different form. But basically, that oocyte's going to sit there. By the time puberty begins, there's only 40,000 of them left. And 400 of them, only 400 will actually wake up to start the race, and we'll talk about the great race in a minute. So these primordial follicles that are hanging out, they really die off like crazy. Carlson says that 99% of the primary follicles are lost during a woman's lifetime. So can you imagine how many babies you could have if they could fix that? Although you could probably are good for about 2 or 3 kids, I would say. I have 5 kids, and I can tell you that I don't know if we could ever do that again in this day and age where things are so expensive. And yeah, so you're probably looking at 2. What's the average? 2.3 kids or something like that. Anyway, so some of these primordial follicles sleep for forever, but some of them start to wake up. So remember, let's see, for the last 12 years, 40,000 primordial follicles are just sleeping. At least 12 years. I mean, some go for 40 years. They're sleeping. But when menarche starts, so when the female starts to mature, probably around the age of 12ish or so, and the first period happens, the great race story begins. So once a month, there is a great race that goes on inside the ovary because there's a chance that one of these primordial follicles will actually mature to the point and start meiosis again, and it can be kind of ejected from the ovary. It's like winning the lottery, probably the odds of that happening. But this great race happens once a month in females, and it alternates. The left ovary goes through it, then the right ovary goes. So every 2 months, one ovary takes a 2-month break. So that's the process of ovulation, and that is the great race, I call it. What wakes up the primordial follicles to start the great race, and how many of these primordial follicles get to be awoken to try the race? It's the authors are all over the place on this again. Some say 5, some say 50. It's probably 20, I would say, on average. So they have some big numbers on these things. But we'll just to keep it, we'll say like 25 of these primordial follicles each month wake up and they start developing like crazy. How do they wake up? Well, now the anterior pituitary gland is involved here. Um, and we're not 100% sure what wakes these things up, but we'll talk about here in a second what that, or maybe we don't. Let me read my slides here. Although pituitary hormones are key for the future development, we're not sure what initiates this group to be awoken. But we do know from this 25 or whatever who wake up, only 3 of them actually have a real chance. So the race begins, and 3 of them are going to kind of come out ahead of the pack and mature. They'll actually kill off the other ones too once they get to a certain size, as we'll see. But out of those 3, only one of them normally will ovulate. Sometimes we haven't talked about twins yet, I don't think. Sometimes you can get 2 of them coming out. So hormones drive their growth. So we know what makes them grow. And when I say race, it's a growth race because the biggest one wins. The one with the most cells in the follicle will kind of win the game. And when these wake up each month, we know that hormones drive their growth. Where do these hormones come from? They come from the anterior pituitary. And here's the little story on that. Really, it's the hypothalamus that starts things off. This is a really important area, and you have to know all the cells in this area quite well. We'll talk about magnocellular neurons, parvocellular neurons when we get in the 5th quarter and how important they are. But for right now, we'll just say there's cells in the hypothalamus that release something called gonadotropin releasing hormone. Gonadotropin releasing hormone, abbreviated GnRH. And that travels down from the hypothalamus to the anterior pituitary, and it stimulates cells in the anterior pituitary to secrete 2 hormones. FSH, follicle stimulating hormone. Oh, that sounds like something we're interested in. And then luteinizing hormone. Together, if you want to talk about these 2 together, these have a name. These are called the gonadotropins. And these are hormones. Hormones, when they're secreted, they jump into the capillaries, get into the bloodstream, and they can stimulate the tip of your nose to your little toe. They can go everywhere in the body, and including the ovary. So FSH is really the big player here. Things can get crazy complicated. We're not going to worry about this right now, but we will when we get in 5th quarter. We might not actually get to this part. Focus more on the adrenal gland. But anyway, so fun facts. So the neurons that make the gonadotropin releasing hormones, this is interesting. So you would think embryologically they would be born in the region of the hypothalamus, but they're not. So this is really weird. They actually are born in the tissue that's going to make up your nose. So they're born in tissue called the nasoplacento. And then in order for them to get into the hypothalamus, they have to travel from the nose tissue all the way into the brain to the hypothalamus. So why am I telling you this? Because there is a disease called Kalman syndrome, and you can see on YouTube some of these people. And they basically never grow up. They're like little kids. And without the gonadotropin releasing hormones, if they fail to migrate, you can never get out of childhood, really, because we need these hormones at the start of puberty to make us into men, the men and women that we are now. And so these Kettleman kids stay kids for a very long time. Maybe they eventually start to mature, but they may be 30-year-old and they're not female, and she doesn't even have her period yet. So that's an interesting syndrome called Kellerman syndrome. They also can't smell. They usually lose their sense of smell. That's all we'll say. We won't go crazy into that. All right, so our follicular cells, which are surrounding the primary oocyte, they have FSH receptors, follicle stimulating hormone. And so what we know about growth and development is that FSH, which is released from the anterior pituitary, floats around the blood, and it finds its receptors on our follicular cells, and it binds to that, and that's what stimulates mitosis to occur, and it causes them to split and grow and grow and grow, and it stimulates growth of the primordial follicle. Binding also stimulates some of these cells in our little follicle here to produce estrogen, which also helps the growth of our little follicle, and we'll just leave it at that. We won't go down that rabbit hole. The first thing that happens, arguably, is so the primordial follicle has changed. Follicles changed to a primary follicle. So we have another name change we need to talk about. Told you this was crazy hard stuff, right? So that's the next step. The primary follicle changes names. So the first thing that happens is a cell type change in the follicular epithelium occurs. So remember, we have these flat cells making up a single layer of the follicular epithelium, or these follicular epithelial cells. So they're going to turn into a cuboidal type change or cell. So we get a metamorphosis into flat into cuboidal. Some authors actually say columnar, but we'll go with cuboidal. Why does that happen? It's thought because of FSH causes a chain of reactions that makes that happen. So it's the binding of FSH to those follicular cells that does that. Primordial follicles. Okay, so arguably, some authors say that once this histological change occurs, so once we've converted our squamous cells into cuboidal cells, we no longer call it the primordial follicle. We call it the primary follicle, or primary growing follicle, some people call it. Now, other authors don't call it the primary follicle yet. They wait until we start, because we have a single layer still surrounding the oocyte. We're still talking about that single layer, that follicular layer. But once it becomes cuboidal for our purposes, then you don't call it the primordial follicle. You call it the primary follicle, or the primary growing follicle. Okay, and remember, we still have this primary oocyte surrounded by a single layer of cells. We haven't done anything more. But now, instead of squamous, they're cuboidal. Now we have a primary follicle. Okay, a lot of words just to make that little change. All right, we're still stuck in prophase 1 of meiosis. Nothing has happened. So not a lot of pictures to put in here, which makes it even harder, but now I got one. So I can show you the primary follicle. So here's our primary oocyte that we've talked about, stuck in prophase 1. And now we've developed cuboidal cells, a layer around it of cuboidal cells. Okay. The only thing wrong with this picture is we haven't talked about this. We have another layer that's just formed here. And we haven't talked about this. We have another layer on the outside. So we need to make a basal lamina, and we need to make something that you might know the words of, the zona pellucida that we talked about last time, because we haven't made those, but we will. Okay, but there's kind of what we have right there. But let's kind of finish the story here. Meet the zona pellucida. So next thing, only the cells that are racing and developing, they develop a new layer immediately around the primary oocyte, and that's called the zona pellucida. Simple as that. But not quite as simple, because we know the zona pellucida has a couple different cell types in it. So the oocyte itself and the follicular cells express genes turned on to make 3 types of proteins that make up the zona pellucida. And so we have ZP1, ZP2, ZP3. That's zona pellucida protein 1, 2, and 3. We talked a little bit about ZP3, didn't we, last time? There's other genes that are turned on. We're not going to worry about those. But ZP3 in particular, there's a star on this slide. This has definitely been asked on several tests. ZP3 is important because it has the receptor to bind the sperm. Remember we talked about that last time. Specifically, sperm has the SED1 receptor, which will hook up and bind to the ZP2 receptor. There's some research that shows the ZP3. SED1 binds to ZP3. There's some research that shows ZP2 might have a secondary binding role to sperm, but they're not 100% sure on that. So for your purposes, the only thing we're worried about is ZP3 is the binding receptor for SED1. I think we looked at this last time. There is the SED1 receptor coming off, much greater. They've really blown this thing up. It's not nearly this big, but it binds to these little blue balls. Those are zona pellucida 3 proteins. Back to the future we went, because we talked about that. SED1 receptors bind to ZP3. Great, easy softball board question. Things like that you can't miss. Communication. So these follicular cells now, we have a layer, right? We have follicular cells, zona pellucida, and now we have our oocyte. So how are you going to communicate with that primary oocyte? You still can communicate with it. There are two types of transzonal projections that penetrate or stick through the zona pellucida and allow the follicular cells to communicate with the oocyte, and they're actually important. So the two that poke through are something called ATZP. So that pokes through, it comes out of the follicular cell and pokes into the microvilli of the plasma membrane. The oocyte, as we'll see next week, has just like the intestinal cells, that does have microvilli on the top of them, and they hook up with there. They use gap junctions, and this is the major route right here, which we'll talk about. This will be important later on. Then there's the MTZP. This allows for paracrine signals to move between cells. Paracrines are like some hormones, can actually not use the blood, and they can just kind of float to their neighbors next door. And that's a paracrine mechanism. So this is the MTZP uses paracrines, or that allows paracrines to move through and get into the oocyte if needed. Not really super important for us, but these guys are important at the end when ovulation is going to occur. All right, now let's get this thing growing. So let's get folliculogenesis going, because we're supposed to be racing here. So the process of a growing follicle, and the goal is to get this out, right? So ovulation is actually the rupturing of this follicle out of the ovary and getting into the fallopian tube. So after the zona pellucida forms, the next thing that happens is the single layer of follicular cells begin to divide through mitosis, and in other words, they proliferate. And so you have one layer, so there's our oocyte, and then we have this follicular layer made of cuboidal cells. So they all undergo mitosis, and now we have, maybe not all at the same time, but now we have a double layer. So follicular layer is now double, and that's what's going to happen again and again and again and again. FSH is thought to stimulate this, and now the race is on. Some are going to grow more layers than others, and we're going to get it down to 3 big ones here in a second, but now the race is on. So folliculogenesis is going. And so as more and more follicular cells are created through mitosis, the follicle, this primary follicle that we have, is going to get bigger and bigger and bigger. And sometimes these, once you get to a double layer, sometimes these are called growing follicles instead of primary follicles. I think we said that already. But that's still an AKA for primary follicle, and growing follicle is still the same. And in the meantime, remember the rest of the 30,000 or so primary oocytes, they're still encased in a primordial follicle. They're sleeping still. So most of them are sleeping until it's their turn to wake up. Okay, the ovarian follicle count. Ross, which is the medical histology book, it's a very, I don't know, June Kira is a good book too. That's the one we use. They're both, you need to read both of them to put the histology story together. But Ross, I use a lot. It points out that there are 3 types of ovarian follicles that can be identified. There's primordial follicles, growing follicles, these are the ones that are racing, and mature follicles. This is where we're going. This is the race right here. We're trying to make 3 mature follicles, and the other ones are going to be killed off. So that's the hierarchy kind of helping us with this race theory. Primordial follicles go to growing follicles, go to mature follicles. Actually, that should be primary follicles. No, that's right, because there's growing follicles, there's 2 types. There's primary follicles are going to turn into secondary follicles. So you could say primordial follicles turn into primary follicles, turn into secondary follicles, turn into mature follicles. The real name for that one is graafian follicles, is what our chiropractic boards use. Whew. I know. I hear your brains going, oh my God, it's way too early for this. But let's go back to the racing story. So we're growing the membrana granulosa. So at this point, we have a multi-layered layers of cells around our little oocyte. And at that point, it's given a new name. It's called the stratum granulosum. Stratum granulosum, that's a multiple layer. At this point, the follicular cells are preferably called granulosa cells instead of follicular cells. The trouble with the embryology authors use follicular cells, and the histology authors use granulosa cells. So you got to remember both of those AKAs. Zona pellucida confusion. I'm a Star Trek fan. Mr. Spock doesn't show emotion right, but he's like confused here. So here's an author problem. Ross says that the zona pellucida forms after the proliferation of the follicle cells, the follicular cells. Larsen, who is an embryology author, says that the zona pellucida has already formed, and we've already said it's formed. So that's a little problem between authors here. And we're going to go with the embryology authors on this one. Nevertheless, the zona pellucida is definitely formed by this point. And then granulosa cells continue to proliferate, and the follicles keeps growing because we're racing, right? Finally, we have a picture of what's going on here. So once this granulosa layer gets about 8 layers thick, a new layer starts to form outside of the original lamina propria. So remember, originally we had 1 layer of cells here, and then a lamina propria, our basement membrane, was on top of these. I think I forgot to say that, but the slide should have said it. There's a lamina propria. Cells always sit on the lamina propria. But as these cells multiply, here's the lamina propria way out here now. And then we have all these follicular cells or granulosa cells filling the region up. So when this gets about 8 layers thick here, another layer of cells is going to start to form out here. And this is going to create a new structure. And some of this is important. Look, we have smooth muscle in here. And that smooth muscle is going to contract and squeeze this oocyte out of the ovary eventually. So this new layer is called the theca folliculi. Theca folliculi. And when does it develop? When the granulosa layer is about 8 cell layers thick. That's when it starts to form. Theca folliculi can actually be split into 2 pieces. A theca interna and a theca externa. So let's look at those a little more closely. The theca interna, and let's take a look at that. So it would be this layer right about here where there's no smooth muscles. I'll color some of it in. So this would be the theca interna. This would be the theca externa out here. Okay. So has a lot of blood vessels. So angiogenesis is the process of forming blood vessels is occurring within the theca interna. There's also fibroblasts and collagen bundles that are filling up this layer as well. There is no smooth muscle here. So that's one of the keys to know if there's no smooth muscle, you're in the theca interna. Smooth muscle is only in the theca externa. Some of those cuboidal cells in there also develop the abilities to secrete some steroid hormones, and we'll just leave it at that. Many of the cells also have luteinizing. We haven't talked about LH, but LH can bind to this layer as well, and I think that's going to be too deep. We're already deep enough into the weeds. Now the theca externa is kind of the star of the show, or it will be in a little while. That smooth muscle fiber has the ability to contract, and that contraction is what helps eject the oocyte. It'll be the secondary oocyte by the time it's ready to eject out of the ovary. And that's what ovulation is all about, ejecting that oocyte that we've been building a wall around. So the next major thing happens when our stratum granulosum here, when it gets to about 12 layers thick, you start getting these little cavities, almost like the same story. Remember the extra embryonic mesoderm, how we develop cavities, and pretty soon the cavities coalesced into the chorionic cavity? Same kind of story here, and it's actually going to create 2 layers. So very similar story. So about 12 layers thick, we develop these fluid-filled cysts, which thank goodness don't have a name, but very quickly they coalesce together into a big cavity, and we're just going to call it the antrum. And no fancy name, so that's pretty easy to remember. Now once you can see under the microscope, once you can see an antrum in one of these follicles, then you know you've reached a new stage. So you no longer have a primary follicle, you have a secondary follicle. Some call it a secondary growing follicle or an antral follicle. They're going to probably call this a secondary follicle. Hyaluronic acid, we will definitely talk about proteoglycans and glycosaminoglycans in spinal anatomy. But hyaluronic acid is actually, this cavity right here is not naked with anything in it. It's got hyaluronic acid, and that has the ability to form proteoglycans, which has the ability to grab water and fill that thing up with a watery kind of gel-like material. See you later. Have a good day. All right, so the fluid inside there has a name, of course, but it's easy. It's the follicular fluid, or some authors call it the liquor folliculi. So watch out for that weird word, liquor folliculi. That's just the fluid that's living inside the antrum here. And this antrum is going to get bigger and bigger, just like the chorionic cavity. And pretty soon it's going to make, just like the chorionic cavity, it's going to leave a stalk. And that's where we're going next. Antral enlargement. So as I said, it grows bigger and bigger, and pretty soon it encircles the entire primary, not quite the entire, but almost the entire primary oocyte. It's going to leave a stalk though, as I just drew, and that stalk is just called the hillock. All right, a lot of author confusion in this area as well. According to Carlson, this antrum causes splits. The development of this cavity causes 2 new layers, just like the chorionic cavity, and so we have 2 new layers to learn. The outer one is called the mural granulosa cell layer. The inner is called the cumulus ophorus cell layer, which is, quite a word, isn't it? Cumulus ophorus cell layer. So now we can see the mural cell layer is on the outside here. And then here is the cumulus orphorus layer. The cumulus orphorus cells also include this little stalk right here as well, which, if our goal is to get this oocyte out and into the fallopian tube, we're actually going to have to break this stalk eventually. And that's what's in store for the future. So the antrum is split into the zona granulosa into 2 layers, a mural granulosa layer and a cumulus ophorus layer. The stalk, or the hillock cells, still is a member of the cumulus ophorus, as I said. Cells immediately surrounding the oocyte will ultimately eject the primary oocyte during ovulation. So we'll look at that in a second. We've talked about the zona pellucida formed, because we talked about that last week, but we also talked about the corona radiata. So these cells are actually renamed, these guys right here, they will actually come with the oocyte. These are the corona radiata cells that we talked about last week. So here's the two barriers, the corona radiata, the zona pellucida. Can't see there's a cell membrane here around the oocyte. But this whole package is what's going to end up in the fallopian tube right here. But we have to just break that stalk down to get that loose. So only one wins the race usually and ovulates. So out of the 50 or 25 or whatever number happens to be starting the race, by this point only 3 of them have usually grown to a very large size, and they're going to be mean and actually kill off the other ones here. But only one of these 3 is actually going to win the race, and the other ones will be lost and degenerate away. How do the big 3 survive? Well, meanly, I guess. They kill the other ones. So the big 3, they eventually, because right now all the growing follicles are dependent on FSH being released from the pituitary. But these big 3, once they get so big, they can actually make their, they don't need FSH anymore from the pituitary. And so they begin to secrete a hormone called inhibin. And inhibin it's a hormone, so it gets in the blood. It can go to your little toe, go to your nose, go to your heart, but where it finds its receptors to bind to is in the pituitary, and we won't get into the exact mechanism, but it shuts off the production of FSH. And without FSH, the big 3 are okay. They can still grow on their own, but the rest of them are still FSH dependent. So the younger ones die off because of the release of inhibin. So that's definitely on the test. I like that little story right there. All right, so now the other ones have died off. We have the big 3 still racing, and now the 24-hour clock starts. Once inhibin is released, we're going to have a winner within 24 hours. So the largest follicle, there's probably one largest by now. Once it reaches a diameter of 10 millimeters, it changes names. So it's no longer a secondary follicle. It's called a graafian follicle, is what most authors call it. Graafian follicle. Some call it a tertiary follicle because really we had a primary, then it morphed into a secondary. So some want to keep it simple and say, okay, this is a tertiary, but for whatever reason they like graafian follicle or mature follicle. I'm going to call it a graafian follicle, but you never know. I might call it a mature follicle, so you should know these. But I usually call it a graafian follicle. So within 24 hours, this graafian follicle will bind to the surface of the ovary. We're still inside the ovary, but it moves up from the inside of the ovary and actually attaches to the surface. And it's going to rupture and release its payload. And its payload is the oocyte with its corona radiata and zona pellucida are going to be squirted out, swept up by the femmurae, and sucked into the fallopian tubes is where we're going. But how does that happen? So around day 14 in the female's menstrual cycle, she releases a massive surge of gonadotropins, both FSH and LSH. That can actually be detected. And that's if you're trying to get pregnant, that's about the time to start working on it. Because ovulation is going to come quickly within a day, within 24 hours. So this big surge of hormones is going to stimulate our new graafian follicle to resume meiosis 1. So finally, things are woke up. Remember, we were stuck in prophase 1, and this surge is going to wake things up, and it's going to start moving through meiosis again. In fact, it's going to finish meiosis 1. Luteinizing hormone. So once luteinizing once luteinizing hormone binds to the cumulus cells, remember we had that flow of, and the question is, what is luteinizing? We haven't talked about that. What does it do? Now it's going to come into the story here. So remember we had a flow of cyclic AMP and GMP kind of being pumped into the nucleus of the oocyte to keep it safe, and that also kind of froze the cell in prophase 1 of meiosis 1. So luteinizing hormone goes in there and it stops that flow. And so, did I say that? Let's see. Yes, right there. I was like, where's stopped? It definitely stops it. Yeah, it stops that flow. That flow is like kind of freezing the oocyte in prophase 1. So luteinizing hormone stops that nonsense. And once that stops and these levels drop inside the nucleus, prophase starts off again, and we go through prophase 1. We go through metaphase, we go through anaphase, we go through telekinesis, and yeah, life is good. We get through prophase. And then ultimately at the end of meiosis 1, we've created a secondary oocyte, and we've also created a polar body, which really doesn't do much of anything. If a sperm binds to that, this whole process was wasted, though. But yeah, that's the story. Meiosis also an important thing because we finish meiosis 1. Remember meiosis 1, just like when we talked about in spermatogenesis, it's a reduction division. So our primary spermatocyte had 46 chromosomes. Our secondary, or not spermatocyte, our primary oocyte had 46 chromosomes. Our secondary oocyte is down to only 23 chromosomes. So its diploid number is 1n, not 2n. Okay, where'd the other chromosomes go? They went into the polar body. They got the other half of the story. All right. So this is where we are. We picked up prophase 1. Once AMP, cyclic AMP, and GMP stopped, this woke up again, and it went right down through here again, and recreated. One of these is a polar body. The author kind of messed that up. But we have one good secondary oocyte, and it actually starts meiosis 2. So that's awesome. The polar body does too. The polar body is going to make 2 more polar bodies. So we're actually ending up with 3 polar bodies and only 1 mature cell, assuming a sperm got in there. But we're going to go through prophase 1 or prophase 2, which is where we got stuck last time in prophase. This time we make it through prophase, but we get stuck in metaphase. So darn it, we're stuck again. Make sure you know for boards and for me where these meiosis 1 gets stuck in oogenesis and where meiosis 2 gets stuck. Okay? Stuck again. So as I said, so little koala bear's got his head stuck between the bars. After going through prophase 2, meiosis 2, the secondary oocyte is stuck again. And this time it's stuck in metaphase 2. And it's going to stay stuck in metaphase 2 forever unless a sperm gets in there and wakes it up for a 3rd time. Or I guess that'd be wake it up for a 2nd time. Okay, let's hatch this thing and be done with this lecture. So about 14 days in, the hillock, remember all those FSH and LSH were released, but the hillock, that little stalk of the cumulus ophorus, it starts to break down because we got to loosen up and get that oocyte ready to squirt out of there. And so that breaks down. The next few hours, the cumuliferous cells start secreting hyaluronic acid, which binds with proteoglycans and absorbs water. So you get a crazy buildup of water within the cell. And this graafian follicle swells like crazy. All right. Because of the water binding, I said that, the expansion, graafian's follicle swells to up to about 20 millimeters in size. And at this size, there's a lot of pressure building up within the cell, right? We have a lot of pressure on the follicle and the ovarian wall. Remember, by this time it's bumped up against the ovarian wall and it's pushing out because it's growing so quickly. And you can actually see on the outside of the ovary, you can actually see this bump developing. Larsen calls this process the cumulus expansion process. All right, and I'm sorry, there's just not a lot of pictures on this story. Let's hatch it. So in order for hatching to occur, that follicular wall, it has to break down, and so we don't have enough pressure yet. Even though that thing is filled with proteoglycans, hyaluronic acid, and everything is swelling like crazy, still not quite enough pressure, and we have to break through the wall to get this thing out. The ovarian wall needs to be broken, and so the theca folliculi, that outer layer, some of the cells start secreting. You would think they'd secrete some enzyme to rip through stuff, but they don't. They secrete histamine and prostaglandin. That's the top of the food chain with regard to the inflammatory cycle. And so that's will cause an inflammation, and the inflammation weakens the ovarian wall and helps it get ready to kind of shoot that, to kind of shoot the graafian, or to shoot the oocyte out, the secondary oocyte out. And at this point you can see a bump, as I said, on the ovary. So here's a real picture of someone about to ovulate that has a name that's called the follicular stigmata. And you can see this over, if we kept following the story, pretty soon this would rip open. And they've moved the fimbriae out of the way so you can see this. But that's called the stigmata, or the follicular stigmata. And that means the race is basically over. We're just waiting for this to kind of rip and shoot its oocyte out into the world so the fimbriae can grab it. Okay, so who finishes off? We got an inflammation, so the ovarian wall. Maybe that's why girls don't feel so good around this time. Sometimes the inflammation can irritate tissue. But the theca follicula, remember, it has that smooth muscle in there. And so the smooth muscle starts to contract, and we won't get into the mechanism because they don't know for sure what causes it to contract. But it knows once that inflammation starts, it's time to start pulling together. And that literally squeezes the secondary oocyte outward. And now the secondary oocyte is broken through the ovary, and it's loose. So the race is over. The secondary oocyte is on the outside of the ovary. Hopefully the fimbriae by now, they can sense this happening, and they move down and they cover, kind of shield the spot where it ruptures. And therefore, normally what happens is once it ruptures, there's all these cilia on the inside of these fimbriae, these fingers, and it's sweeping inward to the lumen of the fallopian tubes. So that little secondary oocyte and its two layers get sucked up into the fallopian tube, and that's what normally happens. If the fimbrium get lazy or for whatever reason, sometimes that oocyte and its zona pellucida and corona radiata, sometimes it gets loose into the pelvic cavity, and you can have an ectopic pregnancy if the fimbriae here of the fallopian tube don't do their job. And we won't go crazy details into this other than what I just said, because we're already overloaded with crazy stuff. But those fimbriae move down over the stigmata. They literally scrape it off there, and they have cilia, and they suck it up into the oviduct, which is nothing more than the lumen, or the inside of the tube of the fallopian tube. And that is the story. Now we got our secondary oocyte inside the fallopian tube. But that's not the end of the story, because now what's going to happen to that follicle, that graafian follicle that we made? Is it wasted? No, it's not wasted. Those granulosa cells that have a hole in it now for kind of shooting the oocyte out of there, they kind of come back to life, and they start secreting hormones. And they get thick, and that hole closes up, and we have a new type of cell. So we don't call them granulosa cells after ovulation occurred. We call them luteal cells. That's another easy question. So after ovulation has occurred, the granulosa cells kind of shift, new genes are expressed and new hormones are secreted, and they become luteal cells, and the graafian follicle also gets renamed at that point. It's called the corpus luteum. It's nothing more than the follicles that we followed, but it gets renamed the corpus luteum once it starts secreting hormones. All right, and that's very important for pregnancy. What does it do? Specifically, it secretes estrogen, progesterone, and those are hormones, so they circulate through the blood. They find the endometrium of the uterus, and they will just leave it at, they prepare it for pregnancy. It thickens, the endometrium thickens in preparation. There's a bunch of stuff that goes on that's too deep. We're already deep enough. If fertilization does occur, or if it fails, if implantation fails, which it does most of the time, the girl goes on to have her period, what happens to the corpus luteum? It degenerates away into a scar-like substance, or little scar bag. And that, of course, has another name for you to memorize. That's called the corpus albicans. Corpus albicans is a worthless structure. It doesn't do anything. What is it? It's the dead corpus luteum. It's called the corpus albicans. If pregnancy occurs, if implantation occurs, the corpus luteum, we'll talk about HCG, how it keeps the corpus luteum alive for much, much longer time than normal. Normally it just turns into this dead scar tissue bag called the corpus albicans. But not if pregnancy occurs. If implantation occurs, HCG will keep the corpus luteum alive for a long time. Whew, that was... Now you know why I said this is probably the most complicated lecture, and good luck to the professor giving this next quarter. All right, let me shut down shop and I'll look to see if there's any questions.